Hi, everybody. I'm Dr. Anna Groat Carmona, and I am an assistant professor in the Sciences and Mathematics Division at the University of Washington, Tacoma. And I am very grateful for the opportunity to talk to you guys about my work looking at the power of gene expression and how it relates to regulating the malaria life cycle. So first things first, malaria is actually transmitted by a eukaryotic pathogen, otherwise known as a parasite. And parasitology is a pretty broad field because it includes everything from ectoparasites like fleas and ticks to endoparasites. And even within this category, we have everything from multicellular worms, which we call helminths, as well as unicellular protists like the one that causes malaria. Now, malaria is what we call a vector-borne disease, and that is because these Plasmodium parasites are transmitted between human hosts via an arthropod vector, in this case, the Anopheline mosquito. Now, while there are over 200 different species of malaria, only 4 are associated with human disease, although a 5th one has recently been identified as a zoonotic cause of malaria. Based on vector distribution, about half of the world's population is at risk for infection. And as recently as 2016, malaria still accounted for over 200 million cases and over 445,000 deaths annually. Now, humans get infected with malaria when infected Anopheline mosquitoes take a blood meal and introduce the Plasmodium sporozoites into the skin. These sporozoites will travel to the liver, where there is an asymptomatic infection that ultimately yields a massive amplification event, resulting in parasitic infection in the blood that leads to the clinical symptoms that you see here. So Plasmodium parasites not only oscillate between mosquito vectors and mammalian hosts, but they also oscillate between invasive and vegetative forms, and even have certain portions of their life cycle where they have sexual forms. These changes are also accompanied by changes in morphology, as well as changes in gene expression. And it is ultimately these changes in gene expression that we want to focus on here. Now, I have a variety of different malaria projects in the lab right now, mainly focusing on liver stage development. But today I actually want to take some time to look at how gene expression dictates the maturation of these sporozoites within the mosquito vector. And so here, these sporozoites are going to be maturing within the midgut lining of these mosquitoes within these structures known as oocysts. Now, within the oocyst, certain genes are going to be turned on, and we refer to these genes as UOS genes, as they are upregulated in oocyst sporozoites. But these genes will be turned off when those sporozoites transition to the salivary glands, where UIS genes are going to be upregulated, as these are upregulated in infectious sporozoites. And these genes are typically associated with those that are going to be necessary for causing disease in the host. Now, in addition to changes in gene expression as you go through the mosquito, you also see differences in their gliding ability, as well as their ability to infect a mammalian host. And this ultimately leads to the question of, well, what's driving this maturation? Changing location in the mosquito, or simply aging within the mosquito? To get at this question, we needed a way to artificially age the sporozoites within the oocysts, effectively preventing them from ever getting to the salivary glands. In order to do that, we actually knocked out an egress cysteine protease from the genome of these malaria parasites, creating the ECP1 knockout. And these parasites simply could not egress from their oocysts. In other words, while mosquitoes infected with wild-type parasites would actually see oocyst sporozoites as well as sporozoites within their salivary glands, these mosquitoes, when infected with ECP1 knockout parasites, well, you just simply never saw those parasites in those salivary glands. They were trapped permanently within those oocysts in the midgut. In addition to being able to trap them in the midgut, because we replaced the ECP1 gene with a GFP cassette that was under the control of a UIS gene promoter, this allowed us to essentially monitor GFP expression as a readout for UIS gene activity. And some interesting things came from this. You see, in the early days of oocyst maturation, where you wouldn't expect those UIS genes to be expressed, well, you don't see any real GFP expression. However, by day 14, despite the fact that these sporozoites are indeed trapped within the midgut oocyst, you do see GFP expression, implying that those UIS genes are indeed being expressed, despite the fact that they are not in the salivary glands. Now, to quantify that pretty picture, we took RNA extracts from wild-type and ECP1 knockout sporozoites at various time points. And what we found is that the UIS genes that you see here were indeed turned on in our wild type, as well as those ECP1 knockout sporozoites. So despite the fact that they were still trapped in the gut, they still expressed UIS genes. In contrast, UOS genes, which should only be on when the sporozoites are in the oocyst, were indeed on at day 11 and turned off for the remaining parts of this assay, and that was true for the variety of different UOS genes that we examined. This implies that these ECP1 knockout sporozoites are indeed undergoing transcriptional changes, and that corresponds to what we would expect to see given the data that's available. So despite the fact that our ECP1 knockout sporozoites are trapped within these oocysts, they are showing gene expression changes that correspond to those sporozoites that are able to go to the salivary glands, meaning there might be some aspects of sporozoite maturation in the mosquito that is time dependent. But to get back to those pretty pictures, we did actually see some interesting things here, because while most of these ECP1 knockout oocysts were GFP positive by day 14 as expected, by day 30 most of them were actually GFP negative, and there's a couple different explanations for why that might be, including protein instability as well as just the death of the sporozoid itself. So there's a lot more to this ECP1 story, as it may have an additional role in sporozoite viability. But deleting ECP1 did allow us to artificially age these sporozoites within those oocysts, and that is a very useful experimental tool to get at this question of age-dependent maturation and looking at really what governs those changes in gene expression within the mosquito vector. Now, believe it or not, changes in gene expression aren't just important for mosquito development. As you'll see from some of my other projects in the lab looking at liver stage development, changes in gene expression can actually have huge consequences on the development of the merozoites that ultimately go on to infect the blood and cause all the symptoms of disease that we call malaria. So with respect to what I'm doing with the liver, a lot of what I do is looking at this Plasmodium BEM46-like protein, or PBLP. PBLP, unlike those UIS and UOS genes that I was telling you about, is actually constitutively expressed and seems to have a global role in the development of invasive stages throughout parasitic development. So now that you know a little bit more about what I've been doing with my time here at UW Tacoma, I want to give a massive shout out to all of my lab members, both past and present, as well as my collaborators at various places in Washington State for all of their great help. So thank you guys all for your time and patience and an opportunity to tell you more about what I do.
Hi, I'm Kelly Kim, and I'm an assistant professor of organic chemistry in my second year here at UW in the Division of Sciences and Mathematics and SIAS. Thank you so much for being here today, and thank you to the Office of Research for giving me this opportunity to share some of the work my lab is doing on synthesizing organic molecules of biological importance. So a lot of us interact with bioactive organic molecules in our everyday life. For instance, if you had a cup of coffee or tea this morning, you enjoy the bioactive properties of the caffeine in it. And so I'm depicting here caffeine in several different ways. You'll notice that organic chemists like to abbreviate because there are so many carbons and hydrogens in many organic compounds that we don't like to draw every single one of them out. So as shown on this slide here, there are much more complex organic molecules than just caffeine. And really they have so many carbon and hydrogen atoms. For instance, each of the points or vertices on these polygonal structures shown here represents a carbon atom that is bonded to other atoms that are either shown or other carbons, as well as hydrogen atoms that are implied. So I'll be using this notation throughout this talk today. And so humans have been able to make use of the bioactive properties of various naturally occurring organic compounds for many centuries, whether it was medicinal purposes, soothing purposes, or maybe even creating a poison. However, it really wasn't until the 1800s, the Volar urea synthesis, that we started to be able to actually target specific organic compounds and synthesize them at will. And so really this makes up a really important skill that we have in our modern society, from preparing materials to agrochemicals and commodity chemicals to creating new medicines and pharmaceuticals, as well as tools to help us study biology and really understand what goes on, the chemistry of life. So it's really this idea that really drives the research that goes on in my lab at UW Tacoma. And really, as a synthetic organic chemistry lab, what we do is we identify interesting targets. If, let's say, they have some interesting biological activity, such as anti-parasitic activity, which I'll talk about today, or maybe some negative property, like some sort of toxicity. And the purpose is really being able to create these compounds and to, in collaborative efforts with biologists, study the biological activities of these compounds. But beyond that, as synthetic chemists, we're also interested in generating new insights, helpful insights into the reactivity of these complex organic scaffolds. And by doing so, inspire the development of new reaction methodologies that'll be useful in other synthetic endeavors. So focusing on the Chagas disease project here, Chagas disease is a neglected tropical disease transmitted via a parasite-infested triatamine bugs endemic to Central and South America. And once in the host, they can cause tissue swelling, heart problems, and GI issues in their human hosts after an acute phase of infection, followed by a really lengthy, decades-long chronic phase of infection. And unfortunately, the drug treatments that are available are really only modestly effective for the acute phase of infection. And for this reason, the Drugs for Neglected Disease Initiative is partnering with academic labs like my own to study some alternative potential therapeutics for this disease. For example, this quinazolinone structure highlighted in purple above, substituted with an amine substituent and a mid substituent, has shown pretty promising activity against the T. cruzi parasite that causes Chagas disease. And so really our first goal here is to establish a robust synthetic pathway to this known bioactive quinazolinone structure for further study, as well as to prepare some derivatives of these disubstituted quinazolinones, for example, varying that amine group and the amide group on the core. And so really as synthetic chemists, we oftentimes start by thinking about what our target is and then kind of thinking backwards, breaking down the molecule into simpler and simpler structures until you get to something that you can purchase from a chemical supplier, and then use your knowledge of organic reactions to stitch them together in a synthesis to get to your final product. So this is the initial synthetic approach we adopted, beginning with the preparation of the chlorinated quinazolinone core, which can then undergo an so-called amination reaction, where you can take an amine functionality that I've highlighted in red, which is the nitrogen-containing group, and if you attach it to the quinazolinone core, you would call that an amination reaction. And so we're able to prepare a number of different aminated quinazolinones shown there. Similarly, attempts to synthesize the bromoamide fragment were also quite successful, and we were able to generate a number of differently substituted bromoamides. And so I'll point out here that the bromine group on these bromoamides was quite important for actually attaching this part of the molecule to the quinazolinone core that I showed previously. So this is where we encountered sort of our first challenge. Of course, in research, it can't be all smooth sailing. We were able to attach the bromoamide to the quinazolinone. However, it did not attach at the desired position. So in the top right of the slide, you'll see that we wanted to attach it at the nitrogen. However, no matter what conditions we tried, it would always attach at the O position, the oxygen. And we know this by looking at the nuclear magnetic resonance spectrum, or NMR spectrum, of these compounds that we acquire at the facilities at UW Seattle. And so the number and size and position and shape of these signals tells us about the connectivity of the atoms in our compounds, and allows us to deduce that what we're actually producing in this reaction is, in fact, this undesired O-alkylation product of the aminated scaffold. So interestingly, if we back up one step and work with an earlier synthetic intermediate, that chloroquinazolinone scaffold, using the same reaction conditions, we are in fact able to successfully N-alkylate our quinazolinone core. So this got us thinking maybe we want to revise our synthetic approach, which is quite common in these synthetic projects. You try one thing, you revise, you try something else, maybe you revise again. So while initially it made sense to break down the compound into kind of two relatively equivalently sized pieces, now with this new evidence, we think it might make sense to put on this amide fragment first and then proceed with the amination. And in fact, this did turn out to be the case, in that the alkylation with all of our different bromoamides that we had created proceeds quite smoothly on our chlorinated scaffold. However, very interestingly, now that we've put on this big amide group, we now no longer can do that previously well-behaved amination. We just get back the starting material, which indicates to us that there's something about the starting material that is causing it to not be reactive. For example, one of the strategies that we're currently exploring is investigating whether perhaps steric effects, or meaning just too much crowding, right? Each of those points is a carbon atom with hydrogens surrounding it. And so perhaps there's just too much crowding around that area for the amine to come in. So we think maybe putting a smaller group and then doing the amination and then building the amide on top of that might work. So to summarize, these efforts are to study the bioactivity of these quinazolinone compounds, perhaps against the T. cruzi parasite that causes Chagas disease, that has limited treatment options despite being a global disease. And along the way, we're learning quite a bit about the reactivity of these quinazolinone compounds. So finally, I'd like to acknowledge the students that did this work. Today, I presented largely the work of Jason Comber and Alex Purcell. So I appreciate all their hard work on this project, as well as the work of the other students in my group who I wasn't able to showcase today. But of course, I appreciate them joining a new lab here at UW and putting their trust in me in that sense. I'd also like to thank our collaborators, as well as all the wonderful staff that makes this research possible. UW Tacoma for their support and funding, as well as you all for your kind attention. Thank you so much.
Hi, I'm Hee-Seok, an assistant professor in new mechanical engineering program at School of Engineering Technology. So today I'm going to talk about a brief overview of my research related to human-powered wearable bioelectronics for healthcare monitoring. Where's just a minute. Okay. So this branch shows my research journey and evolution. So my initial research foundation is in the area of energy conversion, especially direct energy conversion based on thermoelectrics. What is thermoelectrics? If you have N-type and P-type semiconducting material and combine together and apply hot temperature on one end, the other end is kept as cold. And such temperature gradient enable carrier diffusion from hot to cold side. That leads to electric current flow, which is G-bag effect, the principle of solid state power generation. What can thermoelectric do? As we know that Voyager 1 is still traveling, powered by radioisotope thermoelectric generator, which is nuclear reaction in the core to high temperature and capturing cold temperature in outside, and such a temperature difference up to 1,000 degrees Celsius provide electric power to Voyager 1 and still traveling more than 40 years. And it is proved that thermoelectric device is durable. And battery-free LED lights convert candlelight into LED light, which is more useful. And unmanned air vehicle is also interested in using thermoelectric device. As I mentioned, that is thermoelectric is based on solid state power generation. So there is no moving part and very quiet. So it is suitable for military purpose. And more close to this today's talk than body powered devices like flashlight and smartwatch, they are using a temperature difference between body temperature and ambient temperature. And here's an overview of my research on thermoelectrics I've been doing. Material synthesis is focusing on improved material property for higher thermoelectric performance. And device fabrication includes how to measure practical efficiency of thermoelectric system and geometric optimization, how to manage thermal transfer and interfacial study. And there's a huge gap between material technology and device technology on thermoelectrics. So one of my research topic was how to reconcile those two. I'm not going to detail about these today. And on top of that research I enjoyed, I decided to branch out into flexible thermoelectric device for my future career. And this is a typical configuration based on thin film based flexible thermoelectric devices. This is nothing but just make conventional thermoelectric devices and thin and lay down on the plane, which is good. And many fabrication technology has been developed already. But the problem is it is almost impossible to find such a thermal boundary condition, like heat source and heat sink exist at the same plane. It is not realistic. So it is good setup for laboratory setting, but not real application. So I'm working on non-planar flexible thermoelectric device based on 3D structure or self-standing fin-type thermoelectric devices. Then one side is attached to the human skin can as heat source temperature, and opposite side is open to the ambient, even it's very thin configuration.
Hee-Seok,
Yes?
I don't think your slides are forwarding. Where I'm seeing just one slide and it's the tree with solid state conversion thermoelectrics.
Okay, then let me start from here. Is it okay?
Yep.
Okay, so here is a wide range of research field in wearable bioelectronics. And especially I'm working on electrophysiological sensor. And I'm working on a couple of different type of biosensor. And one of them is electrothermal activity monitoring, which is called stress monitoring. So when sympathetic nervous system receive the stressor and they give rise to some signal like sweat gland activity, that leads to skin conductance change. By capturing the skin conductance fluctuation, we can estimate the level of stress activity. And this is the one EDA biosensor Biopatch developed in collaboration with Georgia Tech. And upper right corner figure shows that how to mount that Biopatch on your skin. So we don't need any gel, any adhesive, which is the beauty of this Biopatch. And the lower figure shows that when we remove that Biopatch after a couple of hours using, and nothing left on the skin. So it is said that it is biocompatible or skin compatible. And this graph shows that the measurement result before and after stress activation. So in relaxation period, there's not much peak observed, which means that we don't receive that much stress. And then after the stress activation, we observe lots of peak, means fluctuation of skin conductance. In this case, we use solving a math problem as a strong stressor. And this is concept for continuous monitoring of Biopatch. So by receiving the biopotential signal from the Biopatch and communicate to your smartwatch or smartphone through Bluetooth, and then that data is sharing with your healthcare provider. So this is concept for real-time and continuous healthcare monitoring. But the problem is that, included in my future research, is that handling is sensitive data. So it is critical to incorporate in data security and cryptography. And then I also initiate a different type of research-based self-diagnostics based on AI. Thanks to the many experts in School of Engineering Technology on this data security and cryptography. So I'm going to build a strong collaboration with them and complete this all-in-one type of Biopatch. Thank you for your attention. I'm sorry for about some smooth sharing the screen. So I'm going to stop sharing.
Thank you very much.
This, if you were not aware, is a picture of the former Asarco smelter that's just north of UW campus. You can't quite see campus in the picture, but you can see my house if you look closely. I've been doing active research related to this site for 22 years. So I'm going to cover about 3 years per minute in this talk. And we're going to talk about what threat this historic arsenic contamination in lakes pose for ecosystem and human health. Next slide. All right, I'm collaborating with my co-PI Becca Newman and others shown here over the past years. Also, to meet the teaching and research missions of UW Tacoma, combining the two is a high impact educational practice, and I could not have accomplished this work without the undergraduate researchers that work with me currently and have in the past. So, why should we care? Arsenic is a class A known human carcinogen with myriad cancer and non-cancer health effects. Urban lakes are important sources of food and recreation for many underserved ethnic populations and urban poor, and therefore a human health concern, and possible impacts are there for ecosystem health as well. So the Asarco smelter in Ruston had the tallest emission stack in the world for a very short time, and this helped spread smelter contaminants downwind. The map shows Department of Ecology's predicted deposition zone affected by prevailing winds passing through Puget Sound, and we sampled the lakes that are indicated on the map. We found statistically higher arsenic and lead concentrations in surface lake sediments within the predicted deposition zone, with the highest concentrations in Lake Killarney and Angle Lake up in King County. However, if arsenic remains in the sediments without being transported back into the water, then its potential toxicity to aquatic organisms and humans is low. Killarney is very shallow, 4 meters deep, and Angle is deeper, around 15 meters. So does lake basin depth affect arsenic remobilization? Sediment core data showed that in both lakes, historic arsenic in sediments is being transported up to the sediment water interface, despite the fact that smelter operations ended in the 1980s. First, shallow lakes are warmer at the sediment water interface, almost 10 degrees C in the summer. Light does not penetrate to the bottom of deep lakes, heating only the upper waters. So we found that arsenic dissolved into pore water in the shallow lake sediments was 25 times more than in the deep lake, even with the same amount of arsenic in their solids. Following this with a lab experiment, we took sediment cores from each lake and incubated them at 10 and 20 degrees. At 10 degrees, little arsenic was mobilized from either lake, but at 20 degrees C, the same amount of arsenic was released from both, showing that temperature differences by themselves are a major control on arsenic mobility, and therefore shallow lakes are at risk. Next, warm water is less dense than cool water. Since light penetrates to the bottom of shallow lakes, the temperature difference between the surface and the bottom is small, and thermal stratification of the lake is weak. Therefore, in deep lakes, waters are thermally stratified throughout the summer. Density differences suppress vertical mixing and transport of arsenic. During period of stratification, oxygen is depleted in the bottom waters, a requirement for arsenic mobility. However, periodic mixing events in shallow lakes can break down weakly stratified waters and transport arsenic from bottom waters to oxygenated surface waters. In the presence of oxygen, arsenic eventually adsorbs onto settling particles and is removed from the water. In all lakes, arsenic is bound to fine-grained sediment, which is transported deeper in the lake in a process called sediment focusing. In shallow lakes, unlike deep lakes, the continuous transport of arsenic from deep sediments into surface waters results in the redistribution of arsenic into nearshore sediments. Arsenic concentrations in nearshore sediments of shallow lakes are the same as at depth. Therefore, in deep lakes, arsenic is trapped at depth, restricting the potential for human health impacts on organisms and humans who spend more time in shallow nearshore areas. In shallow lakes, arsenic transport into oxygenated surface waters and nearshore sediments results in spatial overlap between organisms and humans and the contaminant. This then results in uptake into the food chain. Arsenic is chemically similar to phosphorus, a required nutrient for plant growth. Phytoplankton living in surface waters assimilate arsenic at high concentrations in shallow versus deep lakes and transfer the contaminant up the food chain into zooplankton, the first consumers. Arsenic is then transferred into organisms that humans may consume. Chinese mystery snails were introduced into the US as a food source for migrant labor in California. Crayfish and sunfish are both harvestable species in Washington. Arsenic concentrations in their tissues increase with the concentration of arsenic that makes it into shallow oxygenated waters and nearshore sediments. Arsenic does not biomagnify, however, like mercury. Rather, it biodiminishes as it moves up the food chain. In addition, arsenic is transformed into less toxic forms as it moves up the food chain. Therefore, human health risk is higher for those consuming species lower in the food web, like snails and crayfish. Working with the Washington Department of Health, we found increased cancer risks over background for humans consuming these aquatic species. The health risk was highest for and lowest for fish, with highest risk in shallow lakes. Populations who rely more on these sources of food are at particularly high risk, including Native American populations. Although delayed dealing with COVID-19, the Department of Health is considering issuing a consumption advisory for Lake Killarney fish harvest. Even 2 meals a month of fish caught in Lake Killarney would result in a high enough cancer risk to concern the agency. Snails and crayfish have the highest concentrations of arsenic, but it's assumed that fewer people actually harvest these organisms, although we don't know. Applying what we've learned, our future work aims to make it easier for agencies to screen lakes for vulnerability to arsenic human health impacts, develop an affordable remediation technology for arsenic in lake sediments, and understand better the ecological health impacts of arsenic and identify bioindicators for effective monitoring. Current and future work is part of the UW Superfund research program funded by NIEHS. Thanks to my collaborators and our community stakeholders who have supported our work, including the cities of Federal Way and SeaTac, Washington State Departments of Ecology, Health, Natural Resources, and Fish and Wildlife, King County, EPA Region 10, and the Killarney, Steele, North, and Angle Lakes communities. Thank you very much for your time.
It's amazing to see those old pictures of the smokestack.
Yep.
And the changes over in that area.
Mm-hmm. That was the first thing that happened when I moved here is I had my backyard eventually removed because it was contaminated.
My parents live on Commercial Street, so they had that all tested as well. Yeah.
Cool.
So thanks everyone. And today I'm going to talk about this area of research that I've been pursuing for the past few years, studying fairness and bias in machine learning models, along with my work in healthcare artificial intelligence. This talk is really a bittersweet aggregation of things that have worked and things that have not worked. But I'm getting a little ahead of myself here. All right. Do any of you remember this number? Right. And then, the obvious answer might be kind of maybe no, right? How about this, right? It's the same number. How about this one, right? Now with ZB, which is zettabytes. So actually, a few years ago, people were obsessed with this number. It was a projection of the amount of data the world is going to produce in 2025, okay? 175 zettabytes to be exact. And if that wasn't enough, people came up with all sorts of ways to help us visualize this number, this gigantic number. And for some reason, all of these visualizations actually involved DVDs, okay? And it was really, really interesting. In fact, they talked about if you were able to store this data on DVDs, then you could have a stack of DVDs that go from, here to the moon 23 times. And then another one was if you were able to store this data on DVDs, you could have a stack of DVDs that would go around the earth and circle back 222 times. And here's my personal favorite, right? If you could download all that data at an average of about 25 megabytes per second, then it would take one person living in that little apartment in New York just 1.8 billion years to analyze all that data, right? And I love that there is an alternate solution, right? That people can actually help with this. But if every person in the world could help and never rest, then we could just get it done in about 81 days, right? Analyzing this whole data. Where are we going with this? Being impressed by size is a very elegant human imperfection, right? And maybe we can even forgive our younger selves for being so predictably human, since we have then learned that there are many, many other attributes of data that are far more important. Its source, its process, how is it mined, right? How is it obtained? Its integrity, what is missing from this data? How do we know what is missing from this data? Its context, what does the data say about today, yesterday, and tomorrow, right? And so on. But most importantly, for today's discussion and presentation, the biases that are built into this massive data that we are collecting, right? And the algorithms that mine the data sets, petabytes at scale. So what is most important about the data is its characteristic, and it's complicated, right? But it's an exciting future that we face at the same time, but it comes with a profound responsibility that we as computer scientists and everyone here who's doing research have inherited to some extent. Our society has gone through a fundamental digital transformation. If you have been paying attention to the news for the past few years, you may have noticed that the data industry is always in the news. A few years ago, we had massive data breaches. Now we have a bigger problem. And the problem is of algorithmic behavior modification, from election influence interference to automated facial recognition to making people's decisions about who to keep in prisons and just criminal justice and recidivism. Everything is being influenced by biases in the data and in the processes that mine this data. What was once an algorithm that used to decide which movie to recommend on Netflix is the same algorithm now that is helping us to predict, or rather, help physicians predict which medical or clinical pathway to take to treat patients, right? There is this eerie feeling that we have built the, DeLorean time machine, but the wise and measured Doc Brown is not behind the wheel, right? We are time traveling, but without a deeper grasp of the physics of time travel, its foundational axioms, its effect on us, and all our futures, right? So I'll explain this issue of bias through the lens of healthcare. Over the years at UWT, I've worked on numerous predictive models across the continuum of care, from childbirth cost prediction, some of the work that I did with Martine and others, to predicting the risk of mortality at the end of life and everything in between, from risk of readmission predictions to chronic disease management and care recommendations. But as all these predictions started taking shape and we started applying these predictive models into production in health systems across the world, I realized that I'm failing to solve some basic problems in this space. Let me give an example. When we did analysis of data on juvenile diabetes, trying to predict which kids are more likely to deteriorate because of diabetes over the years, we found out that the data tells us that children and young adults with different ethnicities are bound to have different distributions of HbA1c scores or values just within the United States. I'm not even talking globally, right? And any model that ignores this variation will optimize for the ethnicity or the race that is most prevalent in the underlying data set, right? On which it is trained. So not just ethnicity, but age, gender, and even social determinants of health can often lead to significant variation. And AI or artificial intelligence is just not tuned to estimate and eliminate such biases in the predictive models that get put into production today in systems all over the world, right? And it's not just a data problem, it's actually a pipeline problem because we gather and pre process data, we identify imbalances in that data, we build models, we run training and evaluation, we deploy models, make predictions, etc. Right? So the whole plumbing is actually very, very deeply affected by this issue of bias in the system. And there are many sources of bias in artificial intelligence. Most of us only think about it as a data bias problem. It's actually not. There are many non-data bias problems. There are biases in the delivery of the predictive models, and there are several sources of this. For example, the Hawthorne effect is a source of bias where the observed individuals, if they know they are participating in a clinical trial, will actually behave differently when they are on a drug because they are in a clinical trial compared to someone who would take that same drug not being in a clinical trial, right? So there are huge biases. So the question that I'm really asking myself for the next few years as a research question is, how do we operationalize the use of artificial intelligence without regulation and fairness, right? In fact, the question can be rephrased as, how do we usher in the future of AI, or rather responsible AI, right? And there are tons of theoretical frameworks. Some of the work that I've done, many other collaborators have done in publishing these equations and formulations that allow us to measure fairness and bias across different contexts and applications. But I want to highlight that there is this impossibility theorem of fairness, that fairness can never be achieved in any system. And it's a fantastic work by David Levine, and others, where if you try to adjust fairness in one place, it actually breaks somewhere else. So we have to be very mindful about that too. And with that, what I want to go towards and sort of start closing on the talk is 60 to 70% of the projects, all AI projects, fail even after the prediction model has been developed. And you might remember the famous example of Microsoft Clippy, right? That they shipped with Word and then it failed, right? So artificial intelligence in the real world is still a ways away from working very well because the prediction process doesn't get down to the right level of human buy-in because of the issue of trust in AI. And instead of perhaps the question we should be asking ourselves is not how do we go faster from AI research to AI applications, but rather how do we do better going from research to AI applications for the industry and for academia together, right? So towards that goal, I'm now embarking on this journey for the last few years and still continuing the work on every aspect of the AI machine learning pipeline and trying to explore ways where unbiased, features can be found, fairness can be implemented as an algorithmic tool, etc., etc. To bring it all together, I want to end on a happy note and say that your future hasn't been written yet, just like Doc Brown said. No one's has, right? Your future is whatever you make of it. So let's make it a good one. And, hopefully, thank you for the opportunity, Lisa, Cheryl, everyone. And, hopefully I'll get a chance to work with you together. Thank you.
Hi everyone, my name is Martine. I'm a professor at the School of Engineering and Technology. And this year I got a UW Azure Cloud Computing grant through the eScience Institute. And I will talk about how that grant has enabled me to do important research on privacy preserving machine learning with our excellent computer science students at UW. Let's go. So machine learning has grown really big as an area in computer science, and there are many useful and beneficial applications all over the place. For instance, when you apply for a loan or a credit card, you're really putting your faith in the hands of a machine learning system that will help decide whether to approve your application or not. And when you see online advertisements or products being recommended for you, that is because a machine learning system has decided to show those to you. Or when you talk with a virtual personal assistant like Alexa or Siri, and they actually understand you, that's machine learning at work. So whether you are much aware of it or not, we all feel the benefits of machine learning on a daily basis. Now, a challenge is that to work well, these machine learning systems consume a lot of personal data, like our web browsing history, or purchase history, or medical records, and so on. And the need to protect people's privacy is resulting in new laws that restrict what can be done with people's data. And this creates a tension because on one hand, you have all these beautiful machine learning applications that we enjoy to use. And on the other hand, you have new laws that restrict the use of data. And even without laws, we may ourselves feel uncomfortable about disclosing our personal information to companies. So how do we meet this seemingly contradicting need of wanting to protect privacy on one hand and wanting to do or wanting to use private data for machine learning? This is a very important, difficult challenge. And our answer lies in the use of a technique called secure multi-party computation, which allows multiple parties to come together to do computations over data that is encrypted. Now, what kind of computations do we need? Well, in machine learning, many tasks come down to evaluating functions. Internally in computers, all information is represented as numbers. So for instance, when you, as a user, send an email, typically that email is pushed through a spam detector, which is a machine learning model that belongs to some company. For this, first, your email will be converted into a list of numbers called X here, your personal data. And the machine learning model is something you can think of as some complicated mathematical function f. And the system will compute f of x. So the system will evaluate f in the point x and compare it with the threshold. And if f of x exceeds the threshold, say 0.5, a red flag is raised, like, hey, this email is spam. Now, the tricky part is that the decision whether to raise the flag is based on information that is coming from the user X and information F coming from the company. And the user does not want the company to see X, and the company does not want the user to see F, because that machine learning model is part of the secret sauce of the company, right? That helps them to make money. Okay. So our research is about how to compute f of x without either party having to disclose to the other their piece of the input information. To make this happen, we let the user and the company send encrypted shares of their information to servers in the cloud. And the servers compute over this encrypted information to get an encrypted version of f of x. The challenges are, first of all, correctness, making sure that the servers indeed correctly compute f of x and not something else. 2, privacy, making sure that the servers do not learn anything about the real values of x, of f, and f of x. So for the servers, it's really like computing over data that they cannot see. And 3, doing all of this in an efficient manner so that it's actually fast enough to be used in practice. The efficiency is very much influenced by the number of servers used in the cloud. If you have more servers, then you have more compute power, which is a good thing. But at the same time, you will also need to do more communications between the servers, which is going to slow the system down, which is a bad thing. The most exciting part is that when you have more servers, you have more flexibility, more ways in which you can do the encryption and the secret sharing of data between the servers and the way you compute over this. And if you make some smart design choices there, you can really speed up your system. Okay. Now, does all of this work in practice? Yes, it does. We have successfully developed a fast cryptographic protocol for privacy-preserving text classification that can let a company, for instance, detect whether a user's email contains spam without the company needing to look at the email in an unencrypted manner. And our technique can privately classify about 400 words per second, which is very fast, with the state-of-the-art deep learning model that gives you very good accuracy. We obtained this runtime when run in a 2-party computation setting with 2 Azure virtual machines where each of the servers only trusts itself. And we have also developed this year the first secure multi-party computation protocol for privacy-preserving video classification that lets an application, for instance, detect from the video of a user whether the user looks angry or happy or disgusted and so on, without requiring the user to give the application access to their video in an unencrypted manner. And emotion recognition from videos like this is important for the development of empathy-based AI systems. Yet we don't want, of course, these applications to freely collect and view all or of video footage. Depending on the security model that we use and the number of servers, our technique can privately classify a video of 5 seconds in under 14 seconds. This is not fast enough to do real-time privacy-preserving video classification, but hey, we're getting there, we're going in the right direction. We were the first to develop anything like this, right? And we are very excited to see that it can actually work. If there's anything I would like you to remember from this talk is that, yes, we can get the benefits of machine learning without giving up our privacy by using cryptography. And with that, I would like to thank my students and collaborators who worked with me on this research. And I would like to thank Microsoft for donating the cloud computing credits that enabled all these experiments. And thank you all for listening to my talk and staying here until the end in this session.
Thank you, Martine. I think the presentation, it's such a complex topic, but your presentation really made it easier to understand what you're trying to accomplish. So thank you.
Thank you.
Cheryl, do you want to wrap us up?
Sure. So I just want to thank everyone. You want to stop a screen share so we can see our faces, Lisa? That was a really interesting set of topics, all the way from various diseases to body-worn technology, to impacts of arsenic, all the way up the food web to impacting human health if we ingest snails. So the next time I eat escargot, I probably won't eat escargot from Killarney Lake. And then the whole idea of artificial intelligence and machine learning are really hot topics right now, particularly around the bias issue. And that's pretty cool. The bias issue and the privacy issue are huge ones right now. And what's nice about this is I think there's some overlap in the sort of set engineering groups and the biomedical groups that are doing research. And we can kind of share our expertise in doing some interdisciplinary research in the future. And that's kind of the idea of these quickie talks. I know it's not long enough. And I thought Anna and Kelly were going to run out of air because they didn't take any breaths in their slides presentation. But it's really kind of cool to see what everybody's doing. And I think this is a place where we get an idea, ideas about, hey, I'm working on that, or maybe I could apply, that wearable body technology to something I'm studying about with mosquitoes, or maybe I could build in, some organic molecule that would act as an indicator for that. So it's just, I just think, and this is just 6 of us, right? There's a lot more faculty out there, and during COVID, it's so hard to kind of get together and share these things. So I just want to thank you all for presenting, and I'm just, that was kind of fun for me. So and continue sharing, and we'll do more of these next year, and hopefully we'll be back in mostly person in the fall, where we can actually afterwards have a cup of coffee or something and walk out of the room and ask each other questions, and do some more of these when we're not so zoomed out. But thanks again so much.
Thank you, everybody.
All right, you all have a great day.